This is our video on sickle cell anemia for Bio 1510. Our group, Group 9, is consisted of Connor, Jason, Claire, and Maho. To start, we define sickle cell anemia as an autosomal recessive disorder, not sex-linked, passed on by carriers of the sickle cell anemia, and is the most commonly inherited disease in the world. And it is highest in West Africa, where it is found in 25% of the population. Since it is a recessive gene, there are carriers, those affected, and those that are dominantly unaffected. To be a carrier of the allele is a benign condition. It is only in oxygen-low conditions a carrier cells can turn sickle-shaped. Sickle cell is under balancing selection because to have only one copy of the allele protects against malaria. As the host is infected by malaria, the malaria-infected blood cells turn sickle-shaped due to the decreased oxygen tensions of the cell. The body then gets rid of these disease cells at a faster rate than other normal malaria-infected cells. Those affected by the disease may have a difficult daily life, such as, for a child, stunted growth or delayed sexual maturity, along with the swelling of hands and feet, vision problems, and episodes of pain called crises. These pains develop because sickle red blood cells block normal flow of blood, very similar to a blood clot. The irregular and stiff shape of the sickle cells do not allow a normal and smooth flow of blood cells, while normal blood cells are circular and flexible. The purpose of the experiment by Daniel Schreiner and Charles N. Rotem, called whole genome sequence-based halotypes, reveals single origin of the sickle allele during the Holocene wet phase, was to find the origin of this disease within the genome, as well as a geographical and time origin. The approach taken in the experiment was to use data from the whole genome sequence. This included a total of 2,932 individuals from the 1000 Genome Project and the African Genome Project. 156 sickle carriers were found in this population sample. They used a combination of forward time simulation, phylogenetic network analysis, and coalescent analysis to infer a single origin of the sickle allele. The results of the experiment that were found are as follows. The sickle cell mutation was believed to be originated over 7,300 years ago during the Holocene wet phase, either in equatorial Africa or the Middle East. Based on the presence of African ancestry combined with the absence of Arabian or Indian ancestries in the 5 continental African samples in the 1000 Genomes Project, supports an African origin of the sickle mutation. They found that modeling of balancing selection indicated that the heterozygote advantage was 15.2%, an equilibrium frequency of 12% was reached after 87 generations, and the selective environment predated the mutation. This figure was used in the study, and the single branch leading from the ancestral root is the only branch to which the sickle mutation contributes, indicating a single origin of the sickle mutation. These results clarified the origin of the sickle allele and improve and simplify the classification of sickle halotypes. Knowing the origin of the sickle cell mutation provides a wider scope of direction for treatment, including extracting bone marrow, harvesting stem cells, and altering specific genetic instructions to make normal hemoglobin and red blood cells. This is only a treatment, and no cure has been found yet.